This is Victoria Little. I am studying Biomedical Science Foundations, and will be a presentation on the molecular basis of cancer. So cancer is a disease characterized by uninhibited replication of cells. These are cells which are transformed from their normal properties to the extent that they keep dividing even when there are signals independent of the cell that tell them to stop dividing. And they'll keep dividing even beyond the normal boundaries of the tissue. Cancer cells are characterized by this autonomy, as in they are able to divide independent of normal growth signals, and they will proliferate without obeying stop signals. Cancer cells are also characterized by anaplasia. This refers to the fact that there are different types of cells within a tumor or neoplasia because all of these cells are cancerous and therefore have accumulated various mutations. So cells in a tumor are not organized, they're not all the same cell type, they're not making up a tissue towards a certain end or a certain function. It's just a very chaotic clump of cells that keep spreading. So cancer arises from damage to DNA, which sounds simple, but there are a variety of ways that the DNA can become damaged, which we will go into later. Activation of oncogenes and inactivation of tumor suppression genes are the two major pathways that lead to the arising of a cancer. Oncogenes are genes which, when they become activated, will lead to uncontrolled cell growth and survival and proliferation. Tumor suppressor genes, when they are functioning normally, are the body's defense for shutting down any cells that have their DNA damaged and are at risk for becoming cancerous. So naturally, when these tumor suppression genes are inactivated, that puts the cell at increased risk of becoming a uninhibited replicating cancer cell. So as I said earlier, these cancer cells will continue to divide. They will continue to accumulate mutations. They're very genetically unstable, and this actually is pretty beneficial for the cancer cell. It's sort of like a microbiome, like a bacteria or virus. They have very unstable replication cycles with their DNA because it's actually advantageous to accumulate mutation because it'll confer possible mutations that could help the cell survive in a new environment or change the surface of the cancer cell enough that the immune system doesn't recognize it. So mutations are how the cell continues evolving. And another characteristic of cancer cells are they will continue to grow well past the normal boundary of tissue cell to the point where they will even start to spread into different organ systems, far beyond the original primary site of the tumor growth. This is at the expense of other tissues in the body, of course. Not only are these cells invading upon the lining of different tissue types, but they're also essentially sequestering nutrients and starving out these normal, healthy body cells. So there are 6 hallmarks of cancer. We have autonomous growth, which is analogously kind of like stepping on the accelerator pedal, and that accelerator pedal be stuck in the on position. This is a continuous growth that does not stop, which leads into the second hallmark, independence from inhibitory growth factors. Essentially, the brakes for cancer, if we're continuing with this vehicle analogy, are not working and the cell continues to accelerate or divide. Another hallmark is the evasion of apoptosis, or programmed cell death. This means that these cells do not obey external signals trying to kill the cell off for the good of the body. In the normal functioning of the body, if a cell becomes damaged or is infected by an intracellular pathogen, it will secrete certain proteins that visibly mark the cell as damaged and will essentially mark the cell to be killed off. But cancer cells are able to circumvent this pathway. Cancer cells also have limitless replicative potential, so they will have many, many, many progeny cells, and they will accumulate mutations and pass on these abnormal instructions to their progeny. The fifth hallmark is sustained angiogenesis, so cancer cells are actually capable of creating their own blood vessel pathway so that they're able to sequester nutrients and oxygen and even spread throughout the body via the bloodstream by forming their own blood vessel pathways. Though we'll find that these are often pretty faulty blood vessels that they form, not as perfect as normal tissue development. And finally, tissue invasion and metastasis are a hallmark of cancer. This is due to the uncontrolled cell growth. The cancer will actually continue spreading to the point that it goes into adjacent tissues or even escapes into the blood or the lymph vessels to metastasize to an entirely different area of the body. The whole basis of cancer arises from the cell cycle gone kind of horribly wrong. So I think we're all kind of familiar with the normal cell cycle. Starts with, it's not listed on here, but the G0 phase is kind of a little outside cycle phase of a cell cycle that a cell can go through if it is actually removed from the cell cycle and considered dormant. Certain neurons may do this because they're not cells that are going to be rapidly dividing. But for the majority of the body cells, they'll be going through these cell cycles throughout their division cycles. There's the growth 1 phase, where essentially the genetic material is doubling. And then the synthesis phase, where the material actually divides. G2 is when the actual organelles in the cell will double, and then in the mitotic phase, the cell actually divides from one cell into two distinct copies. These little CP red markers here are the checkpoints in each of the cell cycle phases. Usually there is a certain cascade. This is usually jump-started by growth factors, which will cause a cyclin to bind to a CDK, which allows the checkpoint to be bypassed and the cell to move forward in the cell cycle. So it's pretty heavily regulated, which makes sense because we don't want a cell dividing if there's not enough nutrient available. And we don't want a cell dividing when there are plenty of cells around. So there are a lot of external factors that kind of lead into this cascade of, yes, the conditions are good, we can start dividing now as a cell. However, cancer is the uncanny ability of these rampant cells to go rogue and bypass these checkpoints in the cell cycle, so they just continuously keep going through the cell cycle and dividing, despite these checkpoints not being properly bypassed. So one mechanism that this can happen through is through the activation of oncogenes. Right here we're going to talk about two major ones, RAS and MYC. So for RAS, this is a gene that codes for a protein which dwells just on the inside wall of the cell membrane. It's very closely co-located with a growth factor protein, which, a growth factor receptor protein, which whenever a growth factor is released, say when the tissue is trying to signal that it's okay for the cells to start dividing, this will bind into the growth factor receptor, which activates RAS. After RAS is activated, essentially what happens is a series of phosphorylating events take place. Now, phosphate is a very highly charged molecule, so you'll often see it in ATP, GTP. RAS, in fact, itself is a type of GTPase protein. So when RAS is activated, all of these phosphate cascades come down and they eventually end up activating some transcription factors. These transcription factors will go on to transcribe DNA into various proteins which code for CDK and cyclin, which, as we saw on the slide before, actually progress the cell cycle. So essentially what RAS does when it's activated is it causes the cell to progress with its the cell cycle, specifically the checkpoint from G1 into the S phase. A problem when RAS becomes an oncogene is the RAS protein becomes constitutively constitutively activated. So this phosphorylation cascade keeps happening. These transcription factors keep transcribing DNA. And basically the cell keeps dividing and dividing, which, as we know, sounds exactly like cancer. The mycoprotein is a little bit more vague. There's less of a pathway involved with it. It just encodes for all of these different cells. Cellular proteins which are involved with cell growth, cell survival, cell metabolism, which cancer cells have a very high metabolic rate. Alright, on the other hand, we have tumor suppressor genes. Now, the example I'm going to use for this type of gene is P53. So P53, under normal circumstances, is this protein that will actually act as a transcription factor. This protein is expressed whenever DNA is damaged. So, say like there was a mismatch, pinpoint mutation, what have you, and the DNA was copied incorrectly, and for some reason the cell's unable to repair it, or even if like there's some UV radiation that damages the DNA becomes damaged, the cell can't fix it. The cell actually expresses the P53 protein, which will act as a transcription factor to transcribe these P21 proteins. Now, P21 actually goes on to cause cell arrest and DNA repair. So this gives the chance for the cell to actually stop dividing, takes it out of the cell cycle temporarily, and actually gives it a chance to repair all of this DNA damage that was accumulated. And if the DNA is unable to be repaired, that'll signal the cell to go into apoptosis. So obviously, if there is a mutation in this specific tumor suppressor gene, that means there will be no cell arrest happening. The cell will keep dividing even with the DNA being damaged. No DNA repair is taking place, and there's no apoptosis. So you can see how suppression of these TSGs actually leads to cancer development. Alright, now for the origin of cancer, kind of touched on it earlier in that slide, but there are a lot of different ways to damage DNA that can actually lead to cancer. Some common examples are carcinogens, such as smoking. There are a lot of carcinogens in tobacco smoke. There are various radon, nickel are some examples of chemicals that are carcinogens. DNA damage can happen through UV radiation from the sunlight. Various infections are actually able to transform DNA. Some viruses that can cause this include human papillomavirus, Epstein-Barr virus, Hep B and Hep C. And then, of course, there are those risk factors which are heritable, such as the BRCA1 and BRCA2 mutations. These are alleles that are actually confer increased risk for development of ovarian cancer. Okay. There we go. Okay, so now we're going to talk a little bit more about the progression of cancer. So obviously with normal cell growth, you have these cells and they divide when they're told to, obeying, the secretion of growth factors for their specific tissue type. And of course they have their blood supply, so they get adequate oxygen and nutrition, and they stay connected with all the cellular messages in the body, hormones and epicrine signaling. So this is normal cellular functioning. But when a mutation accumulates, there's a possibility of the mutation being problematic enough that it's actually going to induce dysplasic growth. So with this, we'll see a bunch of cells that I colored in purple. These are cells that have been transformed, and that was again through DNA accumulation of mutations, through virus conferring some sort of transformed mutation, or for a carcinogen like tobacco smoke. So this causes some cells to just keep dividing, so they're the cancerous cells. Now, if these cancer cells, as they keep dividing, they'll start to crowd out normal healthy tissue, forming a tumor or a neoplasia. And as the neoplasia kind of gets its feet under itself, it's capable of angiogenesis, which is the forming of its own blood vessel network. So there are a couple of factors which it's capable of secreting, which help form the new blood vessel. One of these is vascular endothelial growth factor alpha, which stimulates migration and proliferation of blood endothelial cells, so the lining of the blood vessels. It's also going to promote vasodilation, which helps to form the lumen of the newly forming blood vessel. Another one is going to be platelet-derived growth factor, which help recruit smooth muscle cell to kind of reinforce the diameter of the larger blood vessels. Angiopoietin 1 and 2 help facilitate maturation of the blood vessels. And then fibroblast growth factor is actually going to draw fibroblastin and macrophages away from the forming blood vessels, so it gets a chance to form without being broken down or engulfed. So as these cells are able to secrete these factors, they're kind of able to form this blood network around them, which helps them sequester oxygen, sequester nutrients, and also kind of has this odd effect of creating areas of inflammation and pockets of hypoxia, which induce mutations, funnily enough, which is actually, again, an advantage for cancer growth, because more mutations are actually a good way of acquiring new abilities for the cancer cell to further evade the body's attempt to clear it out. Another very pertinent example of why having all of this blood vessel access is very good for the growing tumor is that it now has a route to spread to the rest of the body. So the cancer cell can actually break off from the tumor, get into the blood vessel, and go on through the bloodstream to invade the rest of the body. This is called metastasis, if it is able to invade and establish a tumor microenvironment in a new tissue type. Usually the lungs are a common site of secondary tumor growth, because once the cell of the tumor is able to break off into the bloodstream, it'll inevitably wind up in the heart, where it'll go through the pulmonary arteries and eventually set up shop in the lungs. So you'll see that's a pretty common metastatic site for a lot of cancers. And of course, the cancer cells can also grow and invade into surrounding tissues of nearby organs, and of course lymph vessels are also a route that they can spread to other sites in the body. Of course, in the bloodstream, the danger that cancers kind of run into is the immune cells that are circulating. So these are your natural killer cells, your macrophages, the antibodies that might be able to bind to and recognize anything abnormal on the surface of the cell. So for the cancer cell to actually be able to proliferate, get into the bloodstream, and set up shop at a new site in the body, it's got to either avoid immunosurveillance, or it's got to be able to survive whatever the immune system can throw at it. So those are some of the challenges of going into the bloodstream and challenging the rest of the body. So I touched on it a little bit with that previous slide, but the immune response to cancer, I would say the majority of the cell's normal functioning does help to combat tumor growth. Some of the main players are going to be the cells that are capable of engulfing these abnormal cancer cells. This would be your dendritic cells, your macrophages, your neutrophils, the big phagocytic cells that we have in the immune system. Natural killer cells are a type of lymphoid cell, which are actually specially reprogrammed for fighting off cells of the host body, that is, your human cells that were initially normal. And natural killer cells are actually able to take out these cells if they become infected by an intracellular pathogen, or if they become cancerous and stop functioning as they normally should for the body. And then, of course, there are the lymphoid cells, which would be our T lymphocytes and our B lymphocytes. Now, T cells have two special differentiations. Our CD4 T helper cells, which are kind of like generals, which are able to secrete cytokines and all these chemical messengers, which help kind of coordinate an immune response against the cancer cell or pathogen or what have you. And then you have the B cells, which kind of help stimulate the growth of certain immune cells and then direct antibody proliferation. Of course, there are certain versions of macrophages and neutrophils and helper cells which are actually pro-tumor growth and inhibit inflammation. So just to retouch on metastasis, cancer is able to travel to other sites in the body. This can be through the bloodstream or lymph vessels or just to tissues in the adjacent area where the tumor is primarily originates from. Once cancer growth is able to establish in the new site it reaches, the cancer is considered to have metastasized. I think I mentioned this earlier already, but the lungs are usually the secondary site of cancer, just because the cancer cell, once it gets into the bloodstream, it'll go through the heart, through those pulmonary arteries, and set up shop in the lungs. So the process is basically migrating from that original tumor, going into the bloodstream or whatever path it takes to go to the rest of the body, surviving its encounter with the immune cells and setting up shop in whatever the new tissue it locates at. And that will be the secondary tumor. So here are some common sites of metastasis. You can see, just glancing through these, usually the common sites of metastases are the adjacent tissues to the primary tumor site, or those tissues like the heart or the lungs or the brain where the bloodstream is just going to zip the cancer cells right into. So some treatment options for cancer. Traditionally, the big three have been chemotherapy and radiation, which will non-specifically target cells. These will take out cells that are rapidly dividing, which obviously includes cancer cells, but also includes some rapidly dividing healthy cells in the body, like hair, skin, blood, and gut. So that's why with chemotherapy and radiation therapy, you'll often see patients with some detrimental side effects such as hair loss, coughing up blood, nausea, vomiting, fatigue, pain. So it's pretty broad spectrum. It is good for systemic disease when the cancer has spread. When the cancer is all in one spot, surgical intervention is actually a good option for localized cancer. Of course, it's hard to tell that the cancer hasn't spread beyond just a primary site, though imaging technique has made this somewhat easier over the past few decades. You'll see further and further back in cancer treatment history, surgeries could get quite horrific because they're trying to excise as much of that cancerous tissue as they could to make sure that there was no cancer cells left to provide a recurrence. So going forward in cancer, the road ahead kind of just looks at improving our ability to detect cancer, which we are looking into genome sequencing and all of these different tools to identify cancer earlier, which is key to starting treatment earlier and ensuring patient survival. There are also a variety of different treatment options for various cancers. We spoke on the big three that have been traditional treatments throughout cancer's history, but there are some more fine-point treatments emerging, such as immunotherapy, utilizing antibodies, immunoglobulins to target cancer cells specifically and spare the rest of the healthy cells in the body, targeting specific biomarkers so that whenever we treat cancers, we can treat the ones that are exhibiting these abnormal measurable molecular patterns, proton therapy, which is a more pinpoint way of targeting cells, And then CRISPR-Cas9 gene editing can help completely remove any genes that might be worrying, or editing any alleles that are going to end up turning into oncogenes down the road. So just refining our ability to detect cancer and then improving the treatment options that we do have are the big keys that we need to improve on going ahead in oncology. And that was my presentation. Thank you.